Okay, let's start. It's noon. Welcome everybody to the 2020 James Steele Lecture. As it has happened this year, everything is different. This is the first ever time that the Steele Lecture has been delivered online. We're actually happy that we can do it. This lecture was scheduled initially in April, and we really wanted to have Dr. Pamer in Houston, but it wasn't to be this year. The Steele Lecture was created in 1992, established at the UT Health School of Public Health by Dr. Dupont, director of the Center for Infectious Diseases, and Dr. Palmer Beasley, the dean of the UT School of Public Health. The lecture was created in recognition of a lifetime of work carried out by James Steele, the father of veterinary public health, in his bi-pronged efforts to establish the field of veterinary public health and to prevent the spread of infectious diseases from food-producing animals to humans through epidemiological studies and disease control measures. With emerging infectious diseases and bioterrorism threats becoming widespread throughout the world, and the knowledge that a majority of these diseases have an animal origin, Steele's work on zoonosis became even more important. His contributions were varied and dealt with public health control measures during years in the US Public Health Services and with training of countless veterinarians, physicians, and public health students in infectious diseases, epidemiology, and zoonosis. He retired in 1983 and was appointed Professor Emeritus. He died on November 10, 2013 at the age of 100. So it's really an honor for us to commemorate Dr. Steele lecture with Dr. Eric Pamer. Before I introduce Dr. Pamer's, we are putting this together with the Center for Antimicrobial Resistant Microbial Genomic, the Gulf Coast Consortium for Antimicrobial Resistant, and also very importantly, the American Society of Microbiology TMC Chapter. And I want to invite Aisha Khan to say some words about the chapter.
Hi, everyone. Thank you so much for being here. My name is Aisha. I'm a postdoctoral fellow, actually, in Dr. Arias's lab and a co-president of the ASM chapter. And really quickly, we have a lot of new initiatives that we launched this year. So we do a lot of community service, teaching microbiology and mentoring young minority kids in Title I schools, so underfunded schools, as well as a lot of initiatives here to focus on improving our collective microbiology networking and collaborating as much as possible. I'm going to drop 2 links in the chat right now. Everything that we do is on our website. So we're launching a new website that has all of our initiatives in one place. And if you're interested in anything, just make sure to check it out, and you can sign up directly from the website. And we'll continue collaborating with GCC and the Center for Antimicrobial Resistance to put on seminars like this. And you can be a part of that too. So thank you. And again, thank you, Dr. Arias, for giving me the opportunity to do this.
Thank you, Aisha. It's a real pleasure to introduce our speaker, Dr. Eric Pamer from the University of Chicago. He is a professor of medicine and is the Donald Steiner Professor and Director of the Duchossois Family Institute. Dr. Pamer received his MD degree from Case Western Reserve University and completed his clinical training in internal medicine and infectious diseases at the University of California, San Diego Medical Center. He was a postdoctoral fellow with Charles Davis at UCSD, Maggie So at Scripps Research Institute, and Michael Vavan at the University of Washington, and then moved to Yale University and established a laboratory focusing on immune responses to microbial infection. In 2000, he moved his laboratory to Memorial Sloan Kettering Cancer Center in New York, where he was Chief of Infectious Diseases, Head of the Division of Subspecialty Medicine, and Director of the Center for Microbes, Inflammation, and Cancer, which focused on the role of the microbiome in the treatment of cancer. In 2019, Dr. Pamer moved to the University of Chicago to become the Director of the Duchossois Family Institute. His research focuses on the impact of intestinal microbiota in infections in patients receiving cancer treatment, with a special focus on highly immunocompromised patients undergoing allogeneic hematopoietic stem cell transplantation. The Pamer laboratory also investigates the role of inflammatory monocytes in defense against infection and the interaction between intestinal microbes and the mammalian immune system. So Dr. Pamer is a world leader in the field and with extraordinary publication record, and really is an honor to hear his lecture. Dr. Pamer, is all yours. You can start sharing your presentation.
Thank you very much, Cesar. It's really wonderful to be able to see you again, and to be able to talk about some of the work we've done over the past decade. So I'm going to share the screen. Good. Does everybody see that?
Yes.
So
Okay.
yeah, just to make sure, if anyone has any question, please put it in the chat, and I'll do it at the end. Okay.
Okay. Great. So there we go. What I'm going to talk about today is, as Cesar mentioned, the defense against a range of pathogenic organisms. And during my almost 2 decades at Sloan Kettering, I became increasingly interested in some of the organisms that we were seeing in our patients there with increasing frequency. So these antibiotic-resistant bugs like vancomycin-resistant Enterococcus, carbapenem-producing resistant Klebsiellas, and Clostridium difficile. And we happened to be, at the time, starting to study the usual bacteria, the normal bacteria that reside in the intestinal tract. And it developed this interest just at the time when next generation sequencing platforms became more generally available, and enabled us to start to look at the microbiomes of our patients. And that then sort of spawned an interest in me as well as in my laboratory about this. And I generally start my talk with this slide, which is something that's now almost 10 years old. It was in a News and Views in Nature that was written when an organism called the ACEL, which is this little flatworm here, had its whole genome sequenced. And the reason anybody was interested in this thing was that it seems to be a metazoan that had not evolved an intestinal tract yet. So it ingests everything through one hole and then excretes all of its waste through the same hole. And it's believed that around 560 million years ago, there was this big evolutionary step among metazoans to have a second hole so that food could come in one hole and waste could exit through another. And that created a tract, obviously, and is believed to have enabled organisms to grow to much, much larger size because they can eat, digest, and excrete all at the same time. And you can see that all the other life forms or animals that are above this are larger, and they all have an intestinal tract. And what I find particularly fascinating is that bacteria that had been on Earth for perhaps 1 billion years or 2 billion years before this, with this evolutionary step among metazoans, now had a new environment to inhabit. And I think the interesting thing to keep in mind is that throughout the evolution of all animals on our planet, over the past 560 million years, the bacteria that reside in the intestine have co-evolved with us and have become dependent on us, and we have become dependent on them. And this is a huge amount of time for the bacteria to have adapted to us, as well as for us to have adapted to them. So the idea that the bugs that live within our intestines could impact our health, in particular our resistance to diseases, is an old one. It goes way back to Eli Mechnikov, and in fact, over almost 60 years ago now, Rolf Rader published reviews on this topic and already mentioned the ability to control bacterial flora of patients could be of great value. So this concept that the microbiome can enhance health is certainly an old one and nothing very new. And this was a study that certainly opened my eyes to the complexity of the bacterial populations that reside within us. And this is from David Relman's group 15 years ago now, demonstrating on the one hand that everybody has complex microbial populations that live in their lower intestinal tract with lots of different species and strains. And I think one of the most interesting things that he uncovered with this study was that 3 healthy volunteers each harbored a very distinct microbiota, and that has held up. And of course, everybody's aware of the Human Microbiome Project that was performed in the US, and then there was a meta-hit project in Europe that looked at the microbiota in healthy individuals and demonstrated again that in particular in the gut, that people can vary dramatically from being 80 or 90% Bacteroides to being 90% gram-positive organisms that are obligate anaerobes like Clostridia. And it was very quickly imagined that these disparities in microbiota composition could impact disease susceptibility and disease resistance. And you're all aware that there have now been thousands of papers written establishing correlations between microbiome composition and susceptibility to obesity, diabetes, autoimmune diseases. And what we're particularly interested in, of course, is susceptibility or resistance to infectious diseases. Now, one of the other things, again, David Relman was early in showing this, is the effect of antibiotics on the human gut microbiota. And here he studied just what administration of ciprofloxacin, a very commonly used antibiotic, the impact it has and the pervasive effects it has on the microbiota, particularly after it's given in multiple courses. And we, in our lab, looked at this in mice treated, in this case, with clindamycin, an antibiotic that is used quite commonly in the hospital and is well known to predispose patients to, for example, infection with Clostridium difficile. And here we looked at 5 mice, and this is a heat map showing all the different species that are very well represented over the course of 21 days in a mouse that is not treated with antibiotics. However, if you treat with a single dose of clindamycin, what we see, gray means that the bacteria could no longer be seen when we did deep 16S sequencing, that many bacteria that are in the Firmicutes phylum and belong to, for example, Ruminococcaceae families and Lactospiraceae families. We'll talk about those a little bit more if you're not very familiar with them. But these are some of the major families of organisms that live in the lower GI tract that have increasingly been associated with producing metabolites that are health promoting. And those are lost. And in fact, over the course of 28 days, did not return. And what happened was, in place of many of these commensal organisms, things blossomed like Enterobacteriaceae and Enterococci. And that is something that we've seen a lot of in our patients, and I'll show you some of that. Now, another way of looking at the bacterial populations in the gut is by fluorescence in situ hybridization with probes that detect bacteria. And this is a transverse section of a colon of a mouse stained with fluorescent probes that detect bacterial 16S. And what you can see, the surrounding here is the epithelium of the colon, and all of the red are bacteria. and you can see dense populations here of bacteria. And I think what's particularly interesting is that we can see very dense populations of bacteria right here. You can see the epithelial cell nuclei right here, and you can see this dark space in between, and that turns out to be mucin. And that mucin layer is actually protecting us from the bacteria that are residing out there. And they're keeping them perhaps around 50 micrometers away from the epithelial plasma membrane. So when you treat with ampicillin, what you see is you essentially wipe out the bacterial population. So they're much, much less dense. Interestingly, in mice, what you end up getting is a lot more fiber. And that's seen here because, in fact, the bacteria that reside in the lower intestinal tract degrade the fiber into smaller polysaccharides and mono and disaccharides that can be absorbed and can be digested by the host. So just to make the point that antibiotic treatment, which we do very routinely in hospitalized patients and in patients with infections, is having this impact on the microbiota that is really quite dramatic. So the role of antibiotics in predisposing to infection is, again, it's an old idea, and it was investigated incidentally at the University of Chicago back in the 1950s and 60s by Marjorie Bonhoeff and C. Philip Miller and Bill Martin. And what they demonstrated way back then was that if you gave a mouse a single dose of streptomycin, you could make it 1,000,000-fold more susceptible to salmonella. And what I love is this one sentence here by Marjorie Bonhoeff, who recognized already back in 1964 that the most consequential impact of antibiotics is the elimination of certain obligate anaerobes. And that is something that the subsequent decades have demonstrated over and over and over again to be the case, that these obligate anaerobes that are residing in our lower intestinal tract are really doing an amazing job at keeping us resistant to infections. So here's the list. I'm sure many of you have seen this list. This came from the CDC just about a year ago now, listing the urgent, serious, and concerning threats. They all sound bad, but these are the most severe threats, and then the serious threats are down here. And I surrounded in red all of the ones that either emerge from or reside in the intestinal tract. And so you can imagine that for every one of these circled organisms here that are now increasingly becoming antibiotic resistant, that the microbiota in the intestine could play a role in enhancing resistance to them. So I'm going to start with a few short stories. These are topics that we have worked on over the last decade and published on. And then we'll move to some other topics. But to start with, I wanted to focus on these carbapenem and ESBL, extended spectrum beta-lactamase, expressing Enterobacteriaceae. So these are E. coli's and Klebsiellas and Enterobacters. For infectious disease clinicians, these are something that we see many times a day and are asked to consult on because we're challenged to treat them because we're running out of antibiotics to treat them with, and we're also dealing with patients who have allergies to many of these remaining antibiotics. And so it's a big, big challenge clinically. So as Cesar mentioned, we had studied bone marrow transplant patients at Sloan Kettering, and we were supported by a U01 for a number of years, and that enabled us to look almost on a daily rate at the microbiota of patients undergoing bone marrow transplantation. So this is one patient and this is another. And what we see is patients come in pre-transplant and they have lots of different colors. It turns out this mustard color and this pink color are the Lachnospiraceae and Ruminococcaceae that we like to see in the lower intestinal tract. These are all fecal sample sequences. And as the patient goes through transplant and the white count plummets and antibiotics are administered, we lose this diversity. We see E. coli emerging now as a easily detectable organism. As it expands, we now see E. coli in the blood, in a blood culture as the patient spikes a fever over here. We see now introduction of meropenem because it's an ESBL E. coli. E. coli disappears from the gut and is replaced by, turns out, vancomycin-resistant Enterococcus, which does not seem to be impacted by meropenem. We have a very similar picture here in another patient, just showing that this isn't a one-time thing, but we see this actually quite commonly. So a question is, why does now all of a sudden the E. coli expand? And that was something that a postdoc in my lab, Matt Sorbara, studied. And I'm not going to drag you through all of the experiments that he did, but I'm just going to show you the conclusion of how antibiotics impact the lower intestinal tract and enable something like Klebsiella pneumoniae. This is a CARBAPENEM-RESISTANT variant to expand. And the first is that when you treat with antibiotics, the pH in the lower intestinal tract rises. It becomes neutral. And the other thing that happens is that short-chain fatty acids in the lower intestinal tract plummet. So butyrate, acetate, and propionate essentially disappear. Now, what Matt was able to show was that a CARBAPENEM-resistant Klebsiella, for example, grows very happily at a neutral pH. That's not surprising. It also grows very well at a low pH at 5.75. It will grow very well in a physiologic mix of butyrate, acetate, and propionate at a neutral pH, but it will not grow if the pH is low and if you have short-chain fatty acids around. And that's because at this lower pH, these short-chain fatty acids get into the cytosol of the bacteria and drop the pH to a level in the bacteria that disables their ability to proliferate. So that turns out to be an important mechanism of keeping the populations of E. coli, Klebsiella, Enterobacter, Proteus down in the lower GI tract. Now, this is a yet to be published metabolomic analysis that we did together with Justin Cross at Sloan Kettering on a patient who underwent bone marrow transplantation, and we measured propionate, butyrate, and acetate in them. And the patient came in, and because they have a lot of mustard-colored and pink bacteria here, they have propionate, butyrate, and acetate production. And those levels, as the patient goes through transplant, plummet to the level of being undetectable, and that's shown here by the blue and purple. So this is a patient who, as they have progressed, happened to have gotten dominated by Enterococcus. But had they been exposed to an ESBL E. coli, they probably would have, or a CARBAPENEM-resistant one, would have gotten equally dominated with that as well. So I'm going to switch now quickly to talk about some a bit more recent work from our lab on Clostridioides difficile. Obviously, I'm sure all of you are quite familiar with this. It's acquired by ingestion of spores. The spores can germinate in the right setting in the gut, particularly in individuals who have been treated with antibiotics before. And in that setting, the vegetative forms can proliferate, produce toxin, which damages the colonic epithelium and leads to a colitis that can vary from being bad to being lethal. So a question that has been asked for decades is, how does the normal microbiota in the gut suppress C. difficile? So how is it that most people who have never been treated with antibiotics can resist infection with C. difficile, but upon treatment, particularly with an antibiotic like clindamycin, they become susceptible? So a few years ago, Charlie Buffy, an MD PhD student in my laboratory, did some analysis on antibiotic-treated mice that turned out to be a very good model for C. difficile, and compared mice that were rendered susceptible to those who remained resistant to C. diff by sequencing their genome, microbiotas, and he was able to find a number of bacteria that were statistically significantly associated with resistance to C. diff. And the top of the list bug was a bug called Clostridium scindens. And so what Charlie did was he put together a consortium of 4 bacteria, shown here, that he administered to antibiotic-treated mice that were very susceptible. And he was able to markedly increase survival after C. diff infection in these, markedly reduce in vivo C. diff growth by 100-fold, and also almost 1,000-fold reduction in the production of toxin in vivo. Now, a number of labs have been working on the mechanism by which this works. This turns out to be really quite a fascinating biology here for C. diff that, of all things, involves bile salts. And what has been known for quite a long time is that C. difficile spores know that they can germinate when they encounter bile salts in the intestinal tract, and in particular primary bile salts. And those of you who culture C. diff know that you put taurocholate into the media, which drives this germination, and then the vegetative cells can grow. What's maybe less well appreciated, but a number of labs, the Sorg lab in Texas and the Sonnenschein lab at Tufts have demonstrated that secondary bile salts are actually very inhibitory to vegetative bacteria in the colon, vegetative C. diff. And the secondary bile salts are produced by commensal organisms in the gut. And in particular, Clostridium scindens is the most famous one for being able to do that. And it just cleaves a hydroxyl group off of primary bile salts, producing the secondary bile salts. And that's a major reason why, with a healthy microbiota, people don't get C. diff colitis, is because these guys, the vegetative forms, can't grow to a density that would produce enough toxin to damage. Now, you treat with antibiotics, these are very susceptible, as I've already demonstrated to you, and the commensal microbiota disappears, secondary bile salt levels plummet, and vegetative forms can grow. Now, we looked, again, in the same patient I showed you before, where I showed you the short-chain fatty acid levels disappear, at what happens to the 2 major secondary bile acids, deoxycholic acid and lithocholic acid. And you can see when the patient came in, these are pink here, meaning that there was quite a lot of these secondary bile salts, but as the patient went through transplant, these levels plummet. And we now know that this patient, for example, is very vulnerable to dense colonization by Klebsiella, but also is very vulnerable to a C. diff infection. Okay, so I'm now going to give you a bit more detailed story of something that's more recent from my lab on vancomycin-resistant Enterococcus. So I know many of you are going to be very aware of this, this pathogen and the difficulties of treating it. And it's something that at Sloan Kettering was the major cause of bacteremia in our bone marrow and transplant patients, as well as in our leukemia patients. It is in particularly high risk in patients receiving broad spectrum antibiotics. I will just mention now anecdotally, at the University of Chicago, we have started looking at our liver transplant patients, and we have now already had the opportunity to longitudinally collect fecal samples from 14 liver transplant patients here. And what is astounding to me is that they are almost indistinguishable from what we saw in our bone marrow transplant patients, with over half of the patients having almost extreme domination of the gut with vancomycin-resistant Enterococcus. So here is one of our earliest studies. We had just learned how to do shotgun or 16S sequencing using next-gen platforms. So this was 10 years ago now. our first 5 patients. And what I'm showing here in bright green is that 2 patients, patients A and B, got what we refer to as intestinal domination by Enterococcus faecium, and that's shown here. It turned out that these 2 patients ended up developing VRE bacteremia downstream of when their gut was dominated by this. And this then led us to wonder what is enabling vancomycin-resistant Enterococcus to essentially take over the gut. So what's missing in these patients? So one of the things that a postdoc in my lab, Carlos Ubeda, wanted to know was once a gut is dominated by VRE, can the reintroduction of a normal microbiota lead to the clearance of VRE? And so here he used a mouse model, ampicillin, you give the mice VRE, and the blue dots show what happens. The VRE achieves very high density and then persists, even once antibiotics are stopped. Something that's very common that we see in our patients, for example, introduction of normal feces from a mouse that had not been treated with antibiotics, we were very pleased to see resulted in a millionfold reduction of VRE colonization of the gut. Took about a week, but it dropped it down. If we could accomplish this in our patients, I'm pretty sure we could pretty much eliminate transmission of VRE within hospitals. What Silvia Caballero wanted to know was which bugs are doing this. And so this is published a few years ago now. She diluted normal feces from a normal mouse and added the dilutions to mice to see at which dilution do we start to lose the capacity to clear VRE from the gut. And it turned out you could dilute feces a millionfold, 10 to the minus 6 dilution, and you still, most of the mice were able to clear VRE, but there were a few that didn't. And what Sylvia was able to do was to sequence the microbiota composition in these and ask what's missing here, what's underrepresented here, and what's present here that suppresses VRE. And she came up with a list of organisms that, to an infectious disease clinician like myself, were essentially unknowns, guys you hadn't really heard of. Obligate anaerobes of the lower intestinal tract. Now, Sylvia moved on from there and tried to culture many of these different organisms, particularly the ones that were associated with VRE resistance, and she put together a consortium of 4 bugs that we call CBBP, that are these bugs right here. And administering these 4 organisms was as effective as a fecal transplant from a wild-type mouse at reducing VRE in the gut. So we were very excited by that. And Simona Beccatini, in a manuscript that's currently under review, was interested in knowing where these bugs hang out in the lower intestinal tract. And so he used fluorescence in situ hybridization with probes specific to each of these 4 bugs, and found that they, in fact, first of all, they persist for weeks in almost equal proportions in the colon, which is a lucky thing for us. And they seem to commingle in the lumen of the intestine. And we really couldn't see that they either repelled each other or clustered together. They seem to like each other's company and reside in the colon together and persist for weeks on end. We wondered, you know, who's actually clearing the VRE. And so Sean Kim, a MD PhD student in the lab when I was in New York, tested each of the individual bugs in co-cultures with VRE, and what he found, it came out super obvious, was that Blaudia producta completely inhibited, in fact killed VRE upon co-culture in vitro. So we didn't know how it did that, but we had a lot of Blaudia, and we had whole genome sequenced, all of them. And we thought we might be able to do genomic comparisons of all these different Blaudias in order to see what was unique about the Blaudia producta, which inhibits VRE. And so that's this guy right here that inhibits VRE. and all the rest of the Blaudias do not. So that was that was interesting to us. So inhibition of VRE by Blaudia is not a general characteristic of Blaudias. It's something that's specific to this Blaudia producta. So we compared the genomes of all of these, and what Sean was able to do was to fish out a unique and turns out relatively novel operon that encodes something called a lantibiotic. And the structure of the lantibiotic is shown here. This is a very interesting molecule. It's a 33 amino acid protein that is synthesized by ribosomes, by the bacteria. But what distinguishes it from many other things is that it has these lanthionine bonds here between these amino acids. And it's an absolutely fascinating mechanism by which these are generated. And so the genes that are present in this operon dehydrate these amino acids here and introduce these bonds here. This makes this a very stable peptide. It's almost impossible to digest with proteases. And it is incredibly inhibitory to gram-positive organisms. It's very much like a protein called nisin, which is very well characterized and has been around for over 40 years as a food preservative. But this was the first one of these that was found to be produced by an organism that lives and secreted and lives in the colon. And what Sean was able, and I'm not actually showing you a lot of the data that he used to demonstrate this organism, but what he was able to show was that resistance to domination by Enterococcus faecium correlates very much with lantibiotic gene abundance that we were able to detect in fecal samples from bone marrow transplant patients that had been shotgun sequenced. And once you hit a threshold level of lantibiotic gene abundance, we no longer saw expansion of VRE, but below this level we saw lots of it. So I think the point that I wanted to make from these three sort of vignettes between Klebsiella and E. coli and C. difficile and VRE is that colonization resistance is complicated, and it's mediated by a variety of mechanisms. Some, for example, bacteriocin production, which I just talked to you about with antibiotic production, is really a battle between bacteria and can be reproduced in a test tube by just putting the two bacteria together. Many others, however, involve the host. And for example, the ability of Clostridium scindens to modify bile acids so that it inhibits C. diff is something that really is one organism exploiting the host in order to inhibit a pathogenic organism. And there are many other examples of that. For example, commensal organisms inducing immune defenses that drive cytokine production in the lamina propria and production on something we published upon over a decade ago now, antimicrobial factors like Reg3 gamma in the colon. But what we've wanted to do and is to be able to start to move from showing interesting things in mice to being able to actually do something in our patients. A lot has been written, and obviously people are well aware of the fact that fecal transplantation can be very effective at treating recurrent Clostridium difficile infection. But the general utility of fecal transplantation, and I think the desirability of using that in the long term as a treatment, for example, for other antibiotic-resistant infections, I think there are some downsides to it. You know, we never know exactly what's in a fecal sample, even if it's from a healthy donor. And there's variability in the effectiveness of it. And it'd be much more attractive to be able to take specific organisms, put them together as we have in mice, and be able to administer them to humans as a therapeutic. Of course, there are companies out there that are trying to do that. What we wanted to do, and we started this at Sloan Kettering, but it's one of the main reasons I moved to Chicago, is because of the opportunity to do this here and the resources we'll have. And we've established a biobank of commensal strains that come from healthy human donors, and we're growing this now with a real focus on Ruminococcacea and Lactospiraceae and Bacteremia-Mediated, which are the organisms that have been most strongly associated with resistance to infections. Now, we very recently, just a month ago, published a paper focusing just on the Lactospiraceae. So Lactospiraceae encompass the bacteria that produce short-chain fatty acids that suppress E. coli and Klebsiella. They encompass the bacteria that can convert primary to secondary bile acids. And they encompass the bacteria that produce the antibiotics that can suppress VRE. So we focused on these guys, and we wanted to know how diverse they really are. And so here's a tree. These are 273 different isolates. And what we're looking at are genes that they have in common. So all the Lactospiraceae, these are different species here. Only about 20% of the genes that they harbor are shared across the board. And those are mostly housekeeping genes. Some of the genes shown here in the darker grays are shared among the species. So here we have all the Ruminococcus navis, which you may not care about, but it's one of the more common bugs that lives in the intestine and has actually been associated with bowel inflammation. Only about 60% of the genome of all Ruminococcus navis are shared. And they have something here that's shown in light gray and in the dark bluish here, which is referred to as an accessory genome. So that's genomic sequences and genes, whole operons, that are distinct to each strain that is within this species. And this is the area where a lot of, for example, antibiotic resistance, virulence genes, and antibiotic genes are contained. So we've been able to do a deep dive on these Lactospiraceae in terms of their diversity. And here we are using something called a UMAP, which is a way of portraying the diversity in a two-dimensional plot. And the distances between these dots indicates the differences that these have in the number of genes and the types of genes that they contain in their complete genome sequences. So we see certain bugs like Anorostipes hadris clustered together here. Ruminococcus navis actually clusters in a cloud with different subclades here. And then there are a lot of bugs down here that belong to the Blaudia genus that are also clustered very separately from some of the other Lactospiraceae. Now, what this kind of a plot allows us to do is to start to associate the things that contribute to colonization resistance with certain groups of bacteria that belong to the Lactospiraceae family. Here, we're very interested in bugs that lower the pH in the lower intestinal tract because we know that correlates with reduction in the density of antibiotic-resistant E. coli and Klebsiella and Proteus and Enterobacter. And here is where all the acidifiers are. They're all the Blaudias. And we're also interested in butyrate because butyrate is not only immunomodulatory, but it also is very suppressive to Klebsiella and E. coli. And here are all the bugs that produce butyrate. So what we can now see already is that if we want to have a good impact on suppressing Klebsiella or E. coli in the gut, if we just go in with these guys, they're not good acidifiers, so they're not going to be effective. And if we only go in with these guys, they don't produce butyrate, which you need. So in order to actually suppress E. coli, you need some of these and you need some of these guys over here. So that, I think, this is important. We need to, as we start to want to design these organisms, we have to really start to get a good understanding of these bugs and how to assemble them. And this is going to be able to provide a lot of guidance. And I think a lot of what's going on out there right now, particularly by companies that are leaders in this field, is kind of random assembly of organisms to get to the finish line as quickly as possible. But I think these sorts of studies are going to be able to provide a lot of guidance that we need. Here just is another way of sort of breaking down all of these different bugs. We've been digging around in the colon for quite a long time, so we've developed a fascination with bugs like Blaudia wexleri, which essentially all humans have. And now we know that there are within the Blaudia wexleri 3 different clades. And some of them are fascinating and unique in terms of clade A expresses antibiotic immunity genes and also a niacin immunity protein. So this may play a role in suppressing things like VRE or even Listeria, for example, which is very sensitive to antibiotics. Here we see something that's also important, and that is that the metabolic requirements for these different bugs also differs. Here's somebody that likes cellobiose, here's somebody that likes galactonate. So as we start to try to engineer the microbiota in individuals, we're going to have to be aware of what these guys like to eat and what do they like, what fiber do they like to be exposed to. And we can pair that up with dietary supplements or prebiotics in order to enhance their activities. Here, we're looking at Ruminococcus navis, another organism I already mentioned to you. Again, it divides up nicely into 3 clades. Again, that differ in terms of their metabolism, also differ in terms of their antimicrobial that they produce, gramicidins in this case. So a lot to be learned about these kinds of organisms. What I've shown you so far is really only genomic data. But what Matt Sorbara, working together with Jean-Luc Chobard, who now is the head of our metabolomics, we have in the Duchossois Family Institute, we have a microbial metabolomics core facility now with 2 LCMSs and 2 GCMSs dedicated purely to looking for microbial metabolites in both human and mouse samples. And here, what Matt Sorbara did was just to look at metabolite production by our bank of Lactanospiraceae. And what you can see, and actually it turns out it correlates really nicely with the genomic data that we got. So here, Coprococcus comes, big time butyrate producer. Best butyrate producer is Anorostipes hadris. And there are a couple of others. Many other organisms are big time acetate producers, which is good. You'll notice the blue line here is the acetate that's in the media. And it turns out that Anorostipes hadris eats up all the acetate that's in the media. It's part of the biosynthetic pathway it uses to make butyrate. These guys make butyrate in different ways. And so if we ever wanted to use Anorostipes hadris therapeutically, we would probably be smart to pair it up with a big time acetate producer so that it can make butyrate in the right setting. One last complicated plot showing what we can do in terms of characterizing these bugs. We now have a 42 metabolite panel where we can measure not only what do these bugs produce, but also importantly, what do they deplete? And so here we have organisms called Doria longicatena that produce a lot of 5-aminovalerate, which may be important. But in order to do that, it depletes all the serine and proline and cysteine from the media, while other organisms are more selective and only utilize serine. That's what Anorostipes hadris does, and it utilizes up all of the acetate to a big-time butyrate producer. So my point is, why should anybody in public health or in infectious disease care about this? Well, as we're learning more and more, I personally think I would love to be able, on a patient, to be able to get a butyrate level and know what the fecal butyrate level is in a patient, because if it is absent or low, I know that somehow during the course of this patient's medical treatment, their anaerobes have been destroyed, and they would probably benefit from having them repleted. So this is my summary slide. I think I'll still have a little bit of time for questions at the end. I hope that I've been able to convince you that these complex microbial networks provide colonization resistance. The mechanisms, I think we're just scratching the surface. Modification of bile acids is one thing. Secretion of bacteriocins like antibiotics is another. production of short-chain fatty acids, acidification. I think, what I tried to show you towards the end is that some of the bugs that we're wiping out with our piperacillin tazobactam and meropenem, imipenem are these lactospiraceae. They're remarkably diverse. They vary. You can't just pick any bug and call it lactospiraceae and say it's health promoting. We are going to need to know what they have in their accessory genomes. We're going to need to know what they produce in terms of metabolites, and we're going to need to know what they need in order to survive. What partners do they need in order to work effectively? The reconstitution of mucosal bacterial populations can be optimized by careful selecting of these on the basis of genome and metabolic profiles. And then I have a wordy final conclusion here, which is that increasing evidence supports the notion that infectious disease practitioners should probably incorporate the severity of microbiome damage in decision making. So when we choose antibiotics, sometimes we have an option. Sometimes we can think about using an antibiotic where anaerobic infection is very low likelihood, and we can avoid wiping out the anaerobes and still provide good coverage against the organisms we are concerned about. One thing that we talked a lot about with the bone marrow transplanters was switching from piperacillin tazobactam as a go-to drug for febrile neutropenia to cefepime, which is much more preserving of anaerobic populations. And I think what we should do in infectious disease, and I know Cesar is totally on board with this, is that we need to figure out approaches to reestablish health-promoting commensal bacterial populations after antibiotic treatment. So Cesar mentioned that I moved. I came here a year ago to Chicago to lead the Duchossois Family Institute, which is housed in this building, and we're on one small floor here of the building, but we're growing rapidly, and we are recruiting actively, both faculty as well as technicians and workers for our core facilities. And what I want to do is acknowledge my collaborators at Sloan Kettering, Joao Xavier, who is a co-PI with me on the U01 that we had for studying the bone marrow transplant patients. Ying Tor, who is a brilliant infectious disease doctor, as well as epidemiologist and coder, who really made it possible for us to correlate a lot of the microbiome results with the patient outcomes. My long-term friend and collaborator, Marcel Manenbrink, who was on the bone marrow transplant side. Justin Cross, who ran the metabolomics core, and the metabolomics core we have set up at University of Chicago is almost a copy of what Justin has set up at Sloan Kettering. And Sean Kim, who did the antibiotic work. Thomas Moody started the biobank. He's now an MD PhD student at Columbia University, and Peter McKenney has done a lot of our work with VRE and is now an independent investigator at the State University of New York. And then finally, this is Matt Sorbara. Simona Beccattini is now actually an independent investigator in Geneva, Switzerland, and Matt is still with my group. And these are then some of the people that are building some of the core facilities. So with that, I am happy to end. I want to thank you again for your attention and for the honor of being able to speak in the Steele Symposium. So thank you very much.
Thank you, Eric. This was usually humbling and phenomenal. There are more than 110 people in the talk. So you have any questions, please put it in the chat. And I'm going to start with one. I guess there's more. So one of the question is, when would you be ready to administer a cocktail of effective bacterial strains to bone marrow transplant patient? And what kind of regulatory approval would you need for that?
Okay, so that's a huge question. So we are building a GMP facility here so that we can grow on a relatively small scale organisms that we have identified in our mouse models, which we consider a preclinical model, to confer resistance. And we want to be able to assemble these consortia to obtain an IND. I've chatted with people at the FDA. For people in the academic world, the stringency for FDA approval, I think, is somewhat different than it is in the big pharmaceutical companies. Obviously, we'll want to be very careful with the whole thing, but I see us over the course of the next 2 or 3 years being able to move in that direction. Again, working very carefully with the FDA, get approval for our GMP facility, and to be able to manufacture these organisms in a way that could be administered to patients. Now, it could be bone marrow transplant patients. Part of our move here in Chicago has been to identify potential target populations where we have a high suspicion the microbiome is going to be destroyed, and that's why we picked liver transplant, which turns out with treatments that patients undergo for hepatic encephalopathy and for peritonitis, they are exposed to massive amounts of antibiotics, which explains why they almost all are dominated with either E. coli, Klebsiella, or VRE.
Great. Another question is about the short-chain fatty acid pH in Klebsiella. What are the impacts of the microbiota on pH, same or different from microbiota-involved short-chain fatty acid metabolism?
Yeah, so that's a great question, and I probably rushed through that a little bit quickly when I showed my Lactobacteriaceae panel. Maybe I can screen share again. And okay, that's not going to work because I can't, for some reason, I can't go backwards. But what it showed was the big acidifiers were not the ones that were producing the short-chain fatty acids, except for acetate. So they were not producing butyrate, which seems to be a really important player here. So that's seems to be somewhat segregated among these bacteria in terms of who's a big time. The butyrate producers are not great acidifiers. That's the bottom line.
Okay. Regarding the secondary bile acids as inhibitors of C. difficile, ursodeoxycholic acid has been used for a long time in patients with hepatic disorders. Is there any experiment using this medication to prevent toxin production by C. diff?
Yeah, that's an interesting question, and I'm not completely sure about the answer to that. I've been asked this before, and we've never really gotten to the bottom of it. Ursodeoxycholic acid was administered to most of the patients at Sloan Kettering getting bone marrow transplantation, and they still got C. diff. So I'm going to guess that it probably doesn't suppress in the same way that deoxycholic acid and lithocholic acid do.
Okay. Do you foresee treating patients with antibiotics as a drug antibiotic, or is it best to have it produced by the microbiome?
Yeah, I love that question. So I think it's possible that antibiotics may be effective at reducing colonization of the lower GI tract with organisms. In fact, we showed it in mice. If you took niacin, which you can buy, and fed it to mice, you could get a 1 to 2 log decrease in VRE concentrations in the lower GI tract. But that was a small, a very small fraction of what you could do compared to putting in a bacterium that produces it at the site. So as a therapeutic, I think it will be hard to do more than sort of chip away at the concentrations with an orally administered antibiotic, and you would probably have to swallow it multiple times a day in order to achieve that. So, but it does have the advantage, definitely, of not requiring a live biotherapeutic. So what we have sort of thought is that it might be a interesting thing to do in the bone marrow transplant patients during the time when they're neutropenic and you don't want to give them a live bug.
Okay. So the two last questions Can you comment on the use of this type of microbiota intervention on infections by resistant organisms in other organ systems besides the GI tract? For example, UTIs by resistant Klebsiella and other organisms?
Yeah, I think that it's likely that UTIs are probably, in fact, I know that in our renal transplant patients that we looked at together with John Lee at Cornell, that a lot of the UTIs were correlated with expansion of these bugs in the lower GI tract. So I think that would be how one might have an impact on infections in other sites. I think there's a connection between the gut microbiota and the presence of short-chain fatty acid producing organisms in the lower GI tract and resistance to lower respiratory tract infection. We published that. There was a paper published by Bas Hock from our group. And I find that actually quite fascinating. And others, Mike Apt and David Ardis have published a long time ago about germ-free mice having much more severe progression of respiratory tract infections. I don't know that it's really a microbiome in the lung that's driving this. I'm a little skeptical about that, although there are others who believe in that. I suspect that it's more the immunomodulatory effect of these organisms in the lower GI tract and maybe in the small intestine, enhancing immune responses and optimizing immune defenses in other body sites.
Okay, and the last, I know we have the class for our T32 will start as soon as we finish this, but I wanted to give the last question to a friend of yours, Dr. Robert Jank. He says, beautiful recent work linking bacterial genes to metabolic function. Have you tried to go in reverse and learn about genes from the metabolomic data, especially given limitations in gene annotation?
Yes, we have. And we're actually working on a manuscript right now. And we've used machine learning algorithms, random forests, to go in both directions. And so we can now very well, using these machine learning algorithms, predict what metabolites a bug is likely to produce on the basis of its genome. But exactly as Rob just asked, we can also look at a metabolic profile and go in reverse and start to identify or at least correlate it with genomic regions. And so I think this is going to be a very rich area to
absolutely.
Yeah.
Eric, thank you very much. We ran out of time. This is a beautiful talk as usual. I'm glad you're settling down in Chicago. I hope to continue seeing you. I hope to have you in Houston at some point and give you.
Yeah, I want to have you in Chicago too. This has been so much fun seeing you and hearing from some of you. It's nicer to give a seminar in an auditorium and actually make eye contact with people.
Yeah, absolutely. But this is the time we're living. Thank you very much, Eric.
Thank you.
Everybody, the T32 seminar will start shortly. Thank you very much.
Okay, bye-bye.